Let's talk about the cell cycle. My name is Mr. Chipman. I teach AP Biology at Murray High School in Murray, Kentucky, and this is my channel that I talk about things like biology. I actually have other videos. I have anatomy physiology videos. I teach a whole class on that. Zoology videos. I teach an honors biology class. I teach AP Biology. I teach lots of things. You should watch those videos. If you like these videos, you should like them. You should subscribe. You should tell me in the videos, hey, I really like your videos down in the comments, and I'll respond to you with a nice message. So do that. That helped me out a lot. Let's talk about the cell cycle, Unit 4. We got a biology test coming up. Let's do this. Cell cycle is the life cycle of the cell. It's made up of 4 parts. See G1, S, G2. These are all comprised of interphase and the M phase, which is commonly called mitosis. G0 we will mention also. But let's talk about the cell cycle. Let's talk about interphase first. Enter, between, phase, phase. This is the cell between, or this is the phase between cell divisions. During interphase, the nucleus is present. You can literally see the nucleus. This is the normal life cycle of the cell. It's broken up into 3 parts. What is interphase? Oh, well, this just shows you also during interphase. Let me explain this to you. When you look at the nucleus, when I say the nucleus is present, you also can't see the DNA really. It's not like in chromosome form, which we're going to see when we get to cell division. It's just in normal there. This is what this is kind of showing you here. During interphase, the DNA is also replicated. One of the common misconceptions about chromosomes in particular is that they always look like this. Well, this is replicated DNA. These are sister chromatids. We'll talk about that again. But this is what DNA normally looks like when it begins to coil up and form chromosomes. And then you have mitosis. Mitosis is literally the division of the nucleus. And so we will talk about mitosis and the 4 phases therein. Mitosis is not interphase. Interphase is not a part of mitosis. And then the last phase of the cell cycle is called cytokinesis. This is the division of the cytoplasm. The one cell becomes 2 cells. Cytokinesis is a final act, whereas these other things are kind of processes. Cytokinesis is a process, but it represents the actual splitting of those 2 cells. Well, let's talk about these in a little more detail. There it is. Let's talk about interphase first. I just completely skipped over interphase. G1 is growth and preparation. Growth. This is a lot of cells actually never get out of G1 phase. They spend their whole life in G1, wondering what it must be like to replicate their DNA and prepare for mitosis, because they maybe don't have a nucleus like red blood cells, or they just stay in G1. But if a cell is going to divide, it's going to have to go through S phase, which is DNA replication. So G1. I always tell my students that the G stands for growth. It doesn't. I believe it stands for gap, but we're just going to pretend it stands for growth because that's what's happening. And then S phase, synthesis of DNA. DNA is replicated at the end of the S phase. You have 2 copies of the DNA. Why would you do that? Because you're planning on dividing the cell, which is why you're going to do G2. G2 is like the final checklist stage. It's making sure everything is ready to go, like you would do before a trip or something. Do we have everything we need? Preparation for mitosis. Once we get through with G2, we enter into mitosis, prophase, metaphase, anaphase, telophase. You've been going over those probably since 7th grade. We're going to talk about each one of those briefly. And then G0. G0 is kind of a rest phase. Mitosis requires a lot of energy of the cell, and G0 is a way to kind of just take a break from it all. And before it starts G1, it will go into G0 phase. Let's talk about the 4 phases of mitosis. Here they are in all their glory. How many worksheets have you done where you had to label them? Bleh. Okay, prophase. Let's talk about prophase. A couple things are going on during prophase. The DNA is going to begin to condense to form chromosomes. The chromatin is what the DNA is called beforehand. It's just like the DNA is wrapped up on these little proteins called histones. Well, during prophase, those histones are going to begin to condense and form chromosomes. The nuclear envelope, the little membrane that goes around the nucleus, is going to begin to disintegrate. The centrioles are going to move toward the poles of the cell, and everything's going to kind of start to show itself to be not normal. So when you're looking at a cell that's in prophase, when the nucleus is all but gone and these chromosomes are just kind of in disorder in the middle, You're not going to see anything like the chromosomes, but you may see these little fibers, or the centrioles, but you may see these little fibers coming out from them. These little fibers are called spindle fibers, and their job is going to pull the other things towards the centrioles. Centrioles are going to kind of act like anchor points, and the spindles are going to be pulling those structures. Metaphase. This is the phase after prophase. It is where the chromosomes are going to line up in the middle. They're going to line up in the middle of the cell or the equator of the cell. Sometimes you'll see that used because a cell is a three-dimensional object, and so you think of it as like our globe. You have the north and south pole, and the chromosomes are going to line up at the equator during metaphase. An important idea here is that these spindle fibers are going to connect to the chromosomes at the centromere. So let's go back up here to this picture that is right up here. So and this chromosome has two pieces. These two pieces are called sister chromatids. They're identical to one another. And at the center, you have this piece called the centromere. And the centromere is going to have a couple of functions, but the primary function is it's going to serve as a connection point for the spindle fibers during mitosis, which is back down here at metaphase. So those are going to begin to connect. Why are they connecting? Well, anaphase. It's going to pull those sister chromatids apart. They're identical, and so both sides theoretically are getting an equal copy of that DNA. It's like little tow ropes. Ana means to separate, like the word anarchy, separate from the rulers. So literally what that word means, but ana separate phase. So this is where the sister chromatids are separated, move toward opposite ends of the cell. And then telophase, the prefix tela or tela means end or finish. And so this is the last phase of cell division. Notice what's happening. It's kind of an undoing of prophase. Nucleus is going to start to reform. The chromosomes are going to start to unform. Centrioles are going to go back and do their old things before they became centrioles. And we still have two cells, though, or we still have one cell. This is still one cell. Telophase is not the end of cell division. It is the end of mitosis in particular, which is the division of the nucleus. Notice the nucleus has been divided. How do we divide the cell? Well, we do cytokinesis. This is where the cytoplasm is actually divided. Two new cells are made. The language that you will see used is you have one parent cell, and from mitosis you'll have two daughter cells. And this is important because when you start talking about meiosis or meiosis, it's going to be different, because you have four cells that are genetically different from one another. After mitosis, you have two cells, and those two cells are genetically identical to one another. Those cells are often called daughter cells. So there's the cell cycle again. Why do I have another picture of the cell cycle? Because I'm going to talk about cell cycle regulation. One of the ways that the cell cycle is regulated is through the series of checkpoints. Think of checkpoints as like when you're driving your car and there's a police officer there and they're stopping everybody that goes through because it's a checkpoint. Sobriety checkpoint or something like that, right? So there's three checkpoints along the way. There is the G1 checkpoint. I love this picture. Just has everything there. You could pause this. Checks for cell size. Is the cell big enough? Do we really need to divide? Is there enough nutrients? Do we have enough nutrients to sustain the division of the cell? Growth factors, all these things. Is there DNA damage? If there is, we need to just blow up this cell, forget it ever happened. G1 checkpoint. Basically, the G1 checkpoint is oftentimes called the point of no return, because the G1 checkpoint represents a time in which that cell is going to say, okay, after this, we're going to make a new copy of the DNA, and we have to make sure everything's good. And so G1 checkpoint, very important. G2 checkpoint, we're getting ready to divide the cell. Do we have everything we need? Is the DNA damaged? Is the DNA replication process complete? Do we have enough bits to make two cells? All that is going to be important. G2 checkpoint. And the M checkpoint, or the M spindle checkpoint, sometimes you'll see this called metaphase, anaphase checkpoint, whatever, are the chromosomes connected to the spindle fibers at the centromere? That is the question that's being asked here. Sometimes you'll see another word associated with that called the kinetochore. I think that's kind of older language. It's not older language, it's still being used. We just don't use it in this class anymore. Basically, is everything hooked up and ready to go so that when we separate, we're not going to have any problems, because you don't want that. And that is those three checkpoints. They help to regulate the cell cycle. Another thing that's very important is the production of proteins called cyclins. and CDK, or cyclin-dependent kinase. Cyclin, which sounds like cycle, is going to be produced in order to initiate the cell cycle. When it binds with this protein called CDK, it becomes something called maturation promotion factor, or MPF. And that the presence of MPF is going to cause the cell to divide. After the cell divides, cyclin is going to be broken down. CDK, or cyclin-dependent kinase, it's an enzyme, right? It's going to stick around. It's going to wait for the next cell cycle because enzymes stick around. The things that they change do not. This is a great example of that. Let's talk about this in another way. This is a picture you see a lot in AP literature because it's showing you mitosis, right? Mitosis is going to happen. When does it happen? Well, it happens when cyclin concentration gets to a certain point, right? And then once it happens, basically once those chromosomes, once the chromosomes actually split apart at the end of metaphase, beginning of anaphase, then CDK activity is going to completely drop off, cyclin is going to completely dissolve, and it's going to start all over again. Here's another picture of that. This is going to show you that cyclin, CDK, they make MPF. There's lots of maturation promotion factors, but this is kind of a generality. But you can see this S phase, cyclin, CDK, or cyclin, CDK get together, make MPF. MPF is going to be building up in the cell, building up, causing the cell to divide. And then once anaphase happens, cyclin is going to be broken down. CDK is going to wait until it sees more cyclin, do the whole thing over again. What if cyclin doesn't go away? Then the cell cycle is going to keep happening. This is not good. It's not good. Sometimes cells will say, hey, this cell's got a problem, it's not doing right, we're going to go ahead and blow it up. Apoptosis, programmed cell division, or programmed cell death is what that's called. But if that doesn't happen, what if those parts aren't working? What if the thing that says go ahead and die doesn't work? Well, then you got unregulated cell growth or a tumor, and this is called cancer. Cancer is the unregulated cell growth, usually because of some sort of genetic mutation that the proteins that are supposed to say stop dividing, or you can't stop dividing, so go and die. Something is broken, and those cells will continue to grow uncontrollably. Hopefully this was helpful. Hopefully you'll find all of my videos helpful for your review for my exam. If you have questions about the content, please ask down below in the comments. Like this video, please subscribe to my channel. Thank you very much for watching.